Chapter 8 covers cellular reproduction. This is, in essence, cells making copies. This can be done by bacteria through a process known as binary fission, or through mitosis or meiosis in the eukaryotic cells. In both of these, the goal is to get the genes from one generation to the next to make your species survive. Prokaryotic cells are bacterial cells that go through binary fission. In binary fission, you're basically doing asexual reproduction. It's a cloning event. The original first, what we call mother cell, begins with a single circular chromosome that consists of about 3,000 genes. This chromosome, or DNA strand, is found in what is known as the nucleoid region. Remember that prokaryotic cells do not contain a nucleus. In order for these cells to replicate or make copies of themselves, they first must replicate their DNA. So from one circular chromosome, they'll copy that and have two circular chromosomes. These are both attached to the underside of the cell membrane. The cell then, through the different stages, simply grows and adds more membrane between those points of attachment. Once the cell has elongated far enough that the two circular strands of chromosome are farther enough apart, it can then just basically build new membrane and pinch inward, resulting in two genetically identical, what we call daughter cells. In eukaryotic cells, our bodies can go through either mitosis or meiosis depending on the cell type, the location, and the ultimate goal. In mitosis, this is where we make exact duplicate copies, kind of like how the prokaryotic cells did it, only we have a lot more complexity since eukaryotic cells are larger and have a lot more DNA. Mitosis is useful for replacing cells that are damaged or to add cells when you need to grow. In meiosis, this special process takes place only in our reproductive organs, such as the testes and the ovaries. In this process, we produce sperm and egg, otherwise known as the sex cells or gametes. Before we dive right into cell division, we first have to talk about the life cycle of a cell. Most cells spend most of their time in a phase known as interphase. During this, cells are behaving as normal cells. If this is a skin cell, its job is to protect and regulate what gets in and out of your body. Perhaps it helps in thermal regulation, or the pigments within it protect you from ultraviolet radiation. So whatever the cell is, interphase is their normal everyday life. Well, at least the first portion of interphase. If you look at the diagram, you'll see that interphase is broken into subcategories. There are G1, S, and G2. In G1, well, that's when the cell is growing and being a normal cell, doing whatever it's designed to do. During S phase, this is when we copy those chromosomes or DNA strands. In G2, we have a final growth and a large preparation period getting ready for the cell division. In most cells, this takes a lengthy amount of time to complete all three of the phases of interphase. Once they are complete, the cell is ready to actually enter into cell division. That could either be mitosis or meiosis. We'll begin with mitosis, the simpler of the two types of division in eukaryotic cells. Recall that in mitosis, we are making genetic identical daughter cells. From the one mother cell, we go through a process of splitting up our DNA or our chromosomes in such a way that each resulting daughter gets a full complementary set. The only way we can do that is that the mother cell must, in that S phase, make a copy of every single strand of chromosome. When it does so, Each of the duplicated chromosomes are consisting of 2 strands now. You'll note those sitting in the middle diagram. Those 2 copies are called sister chromatids. The sisters will be separated during the process of mitosis in specifically a phase called anaphase. Once those are split apart, we can then build a nucleus around each cluster of those chromosomes and then pinch off the cells and make 2 new daughter cells. Let's dive in more specifically and look at the phases of mitosis. In the first phase, called prophase, basically we are condensing the DNA, meaning we're bundling it up tight into smaller packages, and we're dissolving the nuclear membrane. That means your chromosomes are just spilling out into the cytoplasm. Remember, before prophase, we had to copy all of our DNA, so those represent duplicated copies connected together. So in prophase, our nuclear information is released, we've gotten rid of the nucleus, and we build an apparatus, a structure that's going to help us to move those chromosomes, and this is referred to as the spindle apparatus. In metaphase, where our spindles are going to attach to our chromosomes and drag them and line them up so that they're all basically lined up at the middle of the cell. In anaphase, ana, think of that meaning away or apart. The copies or sister chromatids are separated by those spindles, dragging them towards each polar opposite side. Once anaphase is reached its end, in essence, the 2 clusters of chromosomes are joined at the polar ends in such a way that a new nucleus can be assembled around each of those clusters. That is called telophase. In essence, we're doing the opposite of prophase in telophase. So where we had a spindle that was moving all these chromosomes, we get rid of or dismantle the spindle. Where we had dissolved the nuclear membrane, we rebuild them, and we build 2 of them. And those chromosomes that were tightly bundled up will begin to relax and become not quite so bundled. Officially, mitosis ends at telophase. Mitosis really only has prophase, metaphase, anaphase, and telophase. A different sort of overlapping process called cytokinesis occurs, well, kind of dovetailed over telophase. Cytokinesis literally means cell movement. This is the actual separating of the cytoplasm and the organelles. So in this case, it would be an animal cell. Notice we're covered in a plasma membrane that we can simply pinch inward since we're flexible. Remember that cytokinesis is not officially a stage of mitosis, but is its own stage that dovetails or overlaps the telophase. You can see in this diagram that filaments, basically long linear proteins, help this cell to contract and pull inward and separate basically the cytoplasm and all the organelles. The resulting 2 daughter cells from this are identical to the mother cell that we started with, except they're typically about half the size. They now will enter that G1 of interphase and begin growing. The previous slide showed how animals simply pinch inward. The pinching creates a groove known as the cleavage furrow. That's how you know that you're looking at an animal cell versus a plant cell or some eukaryotic cell that has a cell wall. Looking in this diagram, you'll see that plants, because of the cell wall that is outside of their plasma membrane, has to be reassembled basically kind of brick by brick. You'll see a beginning wall to being built between the 2 nuclei, and it takes some time before the 2 cells can be walled off. These 2 daughter cells are still identical to each other and to the mother cell, just like in the animal cells. The difference is the cell wall makes it look uniquely different. It's great that our cells can divide and replicate and make copies, and they do so generally in a very regulated way. That way, if you cut yourself, you can replace those cells that you've damaged or lost with new cells. So growth and replacement is an essential part of our lives. Unfortunately, sometimes the checks and balances that tell it when to divide and not to divide can get messed up. If a cell is going uncontrollably through these stages and just dividing constantly, well, in essence, that's cancer, uncontrollable division. You'll notice on this chart that there are several checkpoints. Basically, these are sort of guide points that are regulating if the cell is in the right condition, the right shape, and have the right necessary structures to move to the next step. These checkpoints are typically proteins or steroid hormones, which, if you recall, those are part of the lipids. So lipids and proteins serve as checks to make sure the cell is healthy and correctly being stimulated to go through the division when it needs to, and inhibit it from going further if it does not. A quick note on chromosomal numbers. The number of chromosomes or DNA strands in each cell differs depending on the species we're looking at. In humans, we have a total of 46 chromosomes. It could be a lot more. There are some plants that have over 1,200 chromosomes. Our chromosomes are linear strands, not circular like that bacteria. Each of our chromosomes come in a match set, and the sets are known as homologs or homologous pairs. Our pairs are important when we go into meiosis. For right now, let's just give us some names to describe how much DNA you have, or do you have all of your homologs. An animal or a cell that is diploid, note the prefix di means two, means that you have both homologous pairs. You have the homologous pair. If you're a polyploidy, well, that's a plant thing, and they generally have more than 2 copies of their chromosomes. This is really rare in animals and especially in mammals, as too many copies play havoc to our bodies. Now, what if I have a diploid number of 46, like we have in humans, and I cut that number in half to 23? That would be called your haploid number, with hap meaning half. Haploids are written as 1n, and you should memorize that in humans that means 23. The diploid number is written as 2n, meaning you have 2 copies of every one of these pairs, and for humans that's 46. Lastly, chromosomes come in 2 categories. I know the plot is thickening. Our chromosomes, well, we have 22 pairs that are known as autosomes. And these are the chromosomes that hold all the genes that code for your body, for being a human, and for functioning and keeping alive. One pair, however, carries specific genes that are more aligned or associated with giving us gender, male or female. So a total of 23 pairs, 22 pairs of those are known as autosomes, 1 pair are known as sex chromosomes. Together, they make 23 pairs, or 46 total chromosomes. When a cell is in interphase, and definitely in G1, you can't really see the individual DNA strands or chromosomes. And that's because they're not bundled up. They're just laying all diffused out. The entire nucleus looks like one solid dark structure, and that's because your DNA is in a form known as chromatin. But once we start getting ready for division, we make our copies and embark into prophase, we have a definite coiling and bundling. When we go through that process, the long linear strands are wrapped around proteins called histones, and this is usually when we use the term chromosome. Now, remember that in S phase, we make a copy of every strand of our DNA, because stands for synthesis, that is the synthesis or DNA replicating stage, and we attach the copies together. The two copies are known as the sister chromatids. What I didn't point out, though, is that they're attached in a specific location that we call the centromere. So on the far right-hand side of the slide, you can see one duplicated chromosome consisting of two sister chromatids held together in the centromere. That's a region. All eukaryotic cells that go through mitosis and meiosis start off as diploids. That means they have copies of all of their sets, those 23 pairs. Here we can look deeper into what that looks like. Each side is a sister chromatid, and note that they're connected or attached at the centromere. Together they make up a homologous pair. Now, what does the word homologous mean? It refers to homo meaning same and logos meaning structure. And that's because these two homologous pairs, or homologs, carry the same genetic information. Or rather, they have genes in the same locations. For instance, up in the short arm of this chromosome, we may have the gene for eye pigmentation. But the two homologs might not be coding for the exact same color. To be homologous, they just have to code for eye pigmentation. That's what makes them homologous. So we just reviewed mitosis in eukaryotic cells, and remember that is in essence an asexual reproduction. I mentioned it as a growth and replacement, but it could also be used as a type of reproduction. This is often seen in single-celled eukaryotic cells and even in some animals like aphids. They go through a process known as parthenogenesis, where the female essentially lays eggs that are just duplicates of herself. There's no fertilization with a male. If we're going to do sexual reproduction, well, then we have to embark on meiosis. And in order to do that, we must take this process and vary it slightly. That's because we need to produce gametes that have half as much DNA. That's because when mom and dad's gametes join in a process known as fertilization, we need to return back to the original chromosomal number that we started with. So remember, humans had 46 total chromosomes. Our gametes have to be carrying half that, so that when they fuse, 23 and 23, we return back to the total of 46. Cells that are destined to be germinating or giving rise to our gametes, sperm and egg, are referred to as germ cells. These are in contrast to somatic cells. Somatic cells are all your body cells throughout your body, and these are the cells that go through mitosis. Germ cells are those special cells located in our reproductive organs that are destined to go through meiosis. In meiosis, we're going to cut the DNA in half. We'll go from a diploid parent cell, or mother cell, to 4 haploid daughter cells. That means with every meiotic event, we'll end up with potentially 4 sperm or 4 eggs. Meiosis Meiosis includes many of the similar sounding stages, and that's why it's critical that we actually denote their difference by using numbers or numerical numbers. We'll go through 2 divisions instead of 1. In mitosis, where there was prophase, metaphase, anaphase, and telophase, here we'll do that twice, so we need to denote that it is prophase I or prophase II that we are describing. In the first division, we're basically cutting the DNA in half because we are separating the homologous pairs. In the second division, it is essentially identical to mitosis, only that we have half as much DNA. We are still ending up with haploid cells since that's what we had at the end of the first division. Meiosis I is all about cutting the chromosomal number in half. So let's begin with just focusing on the prophase and some of the unique differences that happen in prophase I of meiosis compared to prophase of mitosis. In prophase I, we have 3 similar events that take place in prophase of mitosis. 1, the DNA bundles up tightly into these very visible chromosomes. We also dismantle the nuclear membrane. And lastly, we begin to build the spindle apparatus. Those 3 are exactly the same as prophase and mitosis. But what's uniquely different is when those chromosomes come spilling out of that nucleus, the first thing they do out there in the cytoplasm is search for their homologous pair. And it is these 2 lining side by side, once they find their mate, that an event can take place called crossing over. So this only takes place in prophase I and does not occur in mitosis. In this slide, you can see the crossing over a little bit more in detail. Notice it takes place between homologous pairs of chromosomes, not between sister chromatids. If it did, say, between the two tips of the reds, would we even know? Could you tell that red was swapped with red? That's why the definition of crossing over is the relocation or swapping of a few genes, the tips essentially, of the arms of homologous pairs of chromosomes. Now, how often does this happen? It actually, we believe, happens much more often than we ever thought. In a 2018 study, they found that mammals have crossing over rates of typically 1 per chromosome per event. After Prophase I, Metaphase I is critical in lining up the chromosomes correctly, meaning the homologous pairs line up side by side. Remember that in metaphase of mitosis, every individual chromosome would have lined up at the center. This is important because the way in which they line up can yield a random assortment, or what's called independent assortment, of different clusters or arrangements of our parents' chromosomes that are destined into our gametes. Now let me try to describe that a little bit better. In this slide, under Metaphase I, you'll notice that one parent's chromosomes are colored green and the other parents were purple. So let's say your parents gave you each of these copies, with one coming from your maternal side and one from your paternal side. In this slide, all the greens are lined up on the left, but this is a random alignment. It could have been half and half, with some purples on the right and some purples on the left. That random alignment in all your homologous pairs basically gives you new mixtures. Remember, in humans, this would be 23 pairs lined up, and your 23 parental pairs could be randomly left-right, and that means after anaphase, once we've pulled apart the homologous pairs, and end in telophase, we might not have all of your green, or let's say your father's, chromosomes together. We might have a mixture of your mom and dad's. it's critical that you remember that Anaphase I, we split up the homologous pairs. And in telophase, we do exactly like we've done before. Telophase I is very similar to prophase I. We rebuild the nuclear membrane, we dismantle the spindle, and we begin that process of cytokinesis of pinching this off into two separate cells. The second division that meiosis goes through, or meiosis II, has no chromosomal change ultimately because we're only dividing the sister chromatids. In that way, it is almost identical to mitosis. The only thing different is we have half as many chromosomes. So if you note below, the prophase II starts with 2 cells because they're the 2 we ended with meiosis I. In metaphase II, we line up every single chromosome. In anaphase II, we separate the sister chromatids. And in telophase II and cytokinesis, we divide those up into their own nucleus, pinch off the cell, and result in 4 sex cells or gametes, each of which is truly haploid in its number. We can actually view your chromosomal number to see if you have the correct number in a process known as karyotyping. This is often done in older women or women at risk in pregnancy. They often take a sample of what's known as amniotic fluid, which has sloughed off cells from the baby. They can take those cells, lysis them open, and actually look at the genetic makeup or the chromosomal numbers. So in the bottom left, those chromosomes have just spilt out of the nucleus, and then they've been magnified. Then a computer sort of cuts them out and lines them up or finds their mates or their pairs. So the 23 homologous pairs are lined up based on size. The pair number 1 are our longest, all the way down to pair number 22 and 21, which are incredibly tiny. In this case, you'll notice the very last 2 blanks, one is missing. That means, well, there's a mistake, and something happened. We have a mutation. Many of these mutations, especially missing whole chromosomes, could likely To alter the actual number of your chromosomes, well, that's a mutation, and a common one is known as non-disjunction. In non-disjunction, you fail to split up your sister chromatids or your homologous pairs. That means you might have extra copies going in one direction and not enough chromosomes in the other direction. You can see in the slide how in metaphase, if they lined all up and you accidentally didn't separate the sister chromatids, well, one of these cells is going to have 2 copies of the red and the other one's missing a whole copy altogether. This can happen in our cells anywhere in our bodies, but they're especially problematic when it takes place in metaphase I and anaphase I, or even metaphase II or anaphase II, as those will become the gametes that will pass on the chromosomes to the next generation. That means our children will be born with every cell in their body having an incorrect number. Some of the common mistakes or non-disjunction diseases are things like Down syndrome, which is also known as trisomy 21. Tri means 3, so they have 3 copies of the 21st chromosome. That's one of the tiny ones. Edwards syndrome is trisomy 18. Patau syndrome is called trisomy 13. So these are mistakes in those chromosomes, well, the 22 pairs, not the sex ones. But we can also get non-disjunction in those sex chromosomes. Things like XXX syndrome, which is basically having too many X's and you get bar bodies. This really messes up the body, and we'll talk more about genetics in our next unit. We can also have mistakes in the chromosomes themselves. Individual chromosomes could be misplaced, as in non-disjunction lacking one of those cells. And if that's a sex chromosome, well, we could get something like Turner syndrome. This person only has an X and they're missing the other chromosome. Most of the missing chromosomes in the 22 other homologous pairs would be fatal, and so we typically don't see those very often. Mutations can occur in the individual DNA strands as well. We're not going to focus a lot here, but I just wanted to give you some idea that, well, not only can we misplace entire strands, we can actually have mistakes inside the strands. Look at these couple of different categories: deletions, duplications, inversions, and translocations. All of these are mistakes. Either we're adding or deleting sections of the chromosome. An inversion is a switch or a swapping over, and translocation is when a piece of one gets actually tacked onto another, but it's not crossing over because it's not an equal swap. Most of these are very problematic, causing all kinds of diseases and syndromes that are very dangerous to our health.